traditional knowledge and there we go. Sort of the more modern stuff that I use in my lab, and it's always been a fun exercise, and it's a good go between. We don't put one ahead of the other. There's a place for both as we go. So yeah, so we're going to talk about the work we're doing in the Brador together. And then once we get sort of tell you that story, and again, if people want to jump in and ask questions, by all means, to me or to Anita, just like Anne said, feel free to. And then we'll talk a little bit about a course that we offered, we offered at CBU this year. And what the way the course was structured was every week, students would have, there was 2 lecture slots. One would be a faculty member from Cape Breton University, and then a Mi'kmaq knowledge holder each week. So that's how we did it. So a little bit about us. So the stars represent sort of where I live in sort of Westmount, or not too far from the university. And then in the lower star there, the southern part of the island, or the Brador, is where Anita and Lindsay are from, in the community of Potlotek First Nation. And I'm not much of an artist, as you can tell. I didn't know, I was trying to make this a circle, and then I thought, how do I layer it? How do I do this to show the connections? But essentially what we have is, and the story about how we kind of met was because I teach biology, but I also did teach and still teach sometimes courses in a program, Integrative Science, which its focus is to combine Indigenous knowledge and Western science together at the post-secondary level. And so at one point I was moved from the biology department over to Unama'ki College in the Department of Indigenous Studies, because I was teaching in several different Indigenous communities around Nova Scotia. And Lindsay Marshall was my dean. So Lindsay was the dean of Unama'ki College. I wanted to run a fair size laboratory experiment, but I didn't have any space on campus to do it. I was talking to Lindsay one day and said, do you know anywhere where I could do something like this? And he said, well, why don't you come and do it in our community? So I asked him, well, what do you guys have? And because I didn't really know Lindsay that well, because he was newly appointed and I was newly moved over to Unama'ki College. And so he said, well, come here and I'll show you. So I went into his office, and I thought he was going to show me a picture on his computer, like a phone photo type thing. And he looks up Google Earth and he draws in on the satellite image of this 60 by 40 oyster processing plant that Potlotek has and is hoping to be using very soon. And then he said, you need to go talk to Anita Basque. She's the fishery co-op manager in our community. So I went and banged on Anita's door, and I'm going to guess Anita 15, 16 years ago, give or take. Introduced myself and said, look, I know you guys, have these facilities, and, I'm working on my PhD, trying to do some experiments, could we do something together? And we've been sort of working on this together for quite a while now. So like I said, a good 15 years. And I think it's been really productive. It's been we're making some nice progress, which we'll talk about in a little bit as we go. So that's how we all kind of came to be as a group. I'm just going to give you a quick little bit about this parasite itself, and then I'm going to pass it over to Anita to talk a little bit. So the parasite is Haplosporidium nelsoni. It's called MSX because when it was first diagnosed or first seen in oysters in the Chesapeake, Delaware Bay areas in the late 50s, it was this multinucleated spherical unknown thing. So they labeled it X. So multinucleated spherical X. To this day, we still don't know the life cycle of it. We're hoping to do some work on that now that we have lots of new tools and techniques. And the Brador Lake provides a really interesting and unique place, because you can go to Morson's Cove and it's not there, and go to Gillis Cove, which is probably less than 500 to 800 meters away, and it's in there. So there's some really neat places and really neat aspects about the Brador Lake that we're hoping allow us to work on this parasite together to try to figure out the life cycle. Because if we can figure out that, hopefully we can figure out the life cycle for related diseases, and also come up with some new mitigation measures along the way. So that's the short version. This is actually this isn't the Brador. This is actually McDonald's Pond, which I'll talk about in a little bit, to give you an idea of these are live oysters. This is a place where it sort of was one of the inspirations for one of our bigger projects we've worked on together, but I'll yak about that in a bit. This is down here is what the parasite actually looks like. You can see there's one here, there's one there. When you're looking at it, Mike, what eventually happens is this parasite replicates over and over, and eventually the tissue just completely becomes consumed by the parasite. So all you have is just a large organism with nothing but parasitic cells in it, and then the oyster eventually starves and dies. So this is the movement. It started somewhere around here. down in this Chesapeake, Delaware Bay area, migrated north, migrated south. And then it went as far north as Maine in about 1995 and kind of stagnated there. And then in about we don't know when it came to the Brador actually, but the first mortalities in the Brador were 2001. By 2002, most of the oysters were wiped out on leases and a significant portion of the wild population. So Fisheries and Oceans had no choice but to shut everything down because they had no idea what was going on until they diagnosed what it was, and it was essentially the worst fear. And to some extent, it's largely been stagnated at that point. I'm going to pass it over to Anita a little bit because their community invested and put a lot of effort into preparation for oyster aquaculture.
Hi. Yes, it was devastating. I keep saying like it was a horrible experience for our community. We had an oyster plant just outside the community, about 30 minutes away. And it was difficult to get people to come over, to work. And I decided, and I spoke to my brother, he was involved in the oyster industry too. But anyway, I asked him, we have to somehow get the oyster plant in our community. That way we get a lot of people working and get some oyster leases. So anyway, we got the oyster plant. It's all certified, CFIA. We had buyers and sellers license. But while we were in Louisdale, we were shipping out twice a week. We were shipping all over, like, Portland, Maine, Ontario, Rodney's oyster place. And, we had top of the line oysters. So once we got down to, in our community, we got the oyster plant, and that's what you see in the background. You see some of the elders there, and we collected oysters in some of the leases. And those other people you see over there, that's me over there teaching some of the ladies how to pick oysters and what to look out for. And we're not picking rocks, but we're picking oysters. And some of them said they look like rocks. Oh, they're not rocks, they're oysters. There's actually food inside of these. And but anyway, we've got about 33 oyster leases, applications from our community. Everybody was involved in it, like they were all excited, whatever else, and then MSX hit. And it was devastating. Like I say, people were out of jobs and we couldn't do anymore. We have this huge oyster plant, and we were standstill. So we were just wondering what the heck is going on with this MSX. So we couldn't sell anymore. We couldn't do nothing. All we had to do was just pick some oysters for us and, saute them and make some soups or whatever we had to do. But that's us over there, doing whatever we have to do. But we still continue today.
So just if I'm going to keep on yakking for a bit then. So essentially what happened then is in early 2000, like Anita said, everything gets shut down. We have this confirmation of what it is. But the challenge, of course, the challenge with the Brador is where do we get oysters to bring in? So immediately people were thinking, well, let's bring them in from PEI, New Brunswick, other parts of Nova Scotia. There's lots of oysters from those places. They're not hit by the parasite. But those oysters are all Malpec disease resistant. So Malpec disease hit Malpec Bay and PEI in 1913, 1914, killed almost all the oysters in Malpec Bay, but those that survived are resistant. And to this day, we don't know what causes Malpec disease, but up until I think the last time was done was 96, 97, Fisheries and Oceans put oysters in Malpec Bay from the Brador because they assume that it's never been exposed to the disease agent. And about 18 months after oysters go into Malpec Bay, they die. So they didn't want to bring oysters from outside of the Brador inside, largely because of Malpec disease, but also, and I wasn't in the room at the time, but there were individuals like Charlie Dennis, Jerome Basque, Norman Basque, Mi'kmaq elders saying that we need to work with what's left. We can't the solution is not to bring in things because that's how we got where we are in the first place. So we can't just start dragging things in from other places and putting them in the Brador. So essentially then I kind of left, let nature take its course kind of thing, but that wasn't working very well despite best efforts, largely because it was just going to take too long. Do you have a question, Anne?
I did have a question. Brent asked this question in the chat just while we're talking about kind of the history of MSX. Is it lethal in other oyster species? So do you know if it kills? Yeah.
Yeah. So the only oysters that I'm aware of it's having been detected, no, it's been detected in 3 so far. Pacific oysters, it's not lethal in them. And in fact, that's how it came to this side of North America. It was brought over, in, like I said, the late 50s or so, because the Chesapeake Bay oysters, Delaware Bay oysters were getting fished out. They thought, you know what, we got to replenish this. These Pacific oysters grow much more quickly. Let's bring them in. So they brought them in. Of course, they would have tested them for disease, but back then it was using microscopy. Now, if they go back and look, what they find is that about 10% of Pacific oysters carry this parasite regularly. So they have this sort of tolerance for it. And I can't remember the other oyster that it's been detected in, but we don't see the catastrophic losses that you see. It's not a regular thing in other places. This is very much a North American disease at the moment. So I'm going
to ask a follow-up to that, just while we're learning about it, but we're able to eat oysters that are parasitized with this, so it's not toxic to humans.
Yeah, exactly. So if you were to go down to the United States and eat an oyster, in all likelihood, you're going to need an oyster that's parasitized by one of several different things, but totally harmless to people. So
thank you so much.
So here we are in a situation where oysters can't be taken out of the brador because of MSX, and oysters can't come in the brador because of Malpec. So the brador kind of needs to be its own thing. So one of the things Anita and I have sort of pitched, and we'll talk about some of the research we do together in a few minutes, is this isn't just about the Brador Lake. This is also working to provide using the Brador as a bit of an experiment to learn about what we can do, so that if this parasite shows up in other places in Nova Scotia or in PEI or in New Brunswick, where you have, millions of oysters, hundreds of thousands of, millions of dollars, millions of oysters, and thousands of people working, if we can figure out some solutions here, it provides some, hopefully, degree of insurance or protection for other places. And if you look at that little chart, you can see, I don't know how to move. Let me just move this for a second. There we go. So you can see right here, I keep forgetting you guys can see what I see. This right here is sort of, you can see how oyster production really started. And this is where Potlotek was buying their equipment. And really, you can see oyster production is really, really starting to increase. When the Brador Lake got shut down, this happened, and this represented about 80% of oyster production in Nova Scotia. Basically disappeared in a matter of months because oysters were taken from the Brador and spread throughout Atlantic Canada because it was such a great nursery area. So the current state of the parasite on Cape Breton, because it's the only place in Canada, in addition to being the most beautiful island, it's also the only place in Canada who has this, as far as we know. If we test enough for it, we might find it in other places. I'm not sure testing oysters is exactly the best way to go, but that's a personal opinion. Anyway, but what we do know is the Brador Lake is all considered positive, even though there's places where we've tested for it and haven't found it, but that doesn't mean it's not there. It's just mean we've not detected it. There's this little place in McDonald's Pond. That's that original photo I showed you, just outside Myra Bay, and also up in Aspy Bay. So those sort of 3 places right now have it. It's kind of been undetectable up here for a while. This is a real bizarre phenomena that we talk about, that we see in this place, which I can talk about another time. But what we do know is that oysters up here and here were transferred from the Brador Lake. We know that for a fact. So those are absolute transplants. So, but the reason this is should be a major concern for the region is these are some of the known transfers that are documented transfers from the Brador to other places in Nova Scotia. So, it's just a matter of the right organisms, the right environmental conditions, if the parasite happened to be transferred there as well. There's just another picture. You can see the parasite itself. And if you look at what happens is it gets taken in by the gill, along the gills, and what happens is you'll go from 1 to 2, and then next thing you know, you're seeing tens and hundreds and thousands until the entire animal is nothing but these little spheres. This is some of the work. How do I make this go away? I'm not very good at this. There, that's better. Let me just move you people down there. Okay, yeah. So some of the things we've been able to do is we're able to move the parasite out of out of the oysters. So we can take infected oysters and hold them in tanks, and they naturally are able to purge it somehow. We don't know the mechanisms involved completely yet, but that's a project for another time. That's potentially another use for the facility that they have in Potlotek. Is in addition to a processing facility, it could easily be used as a treatment facility. There was some discussion about people with leases not seeing their oysters dying. So what we did is we moved oysters from their leases into Nyanza Bay, where we know there's significant oyster parasite activity. And what we found was that there's no evidence for resistance to this infection or mortality. It's just some locations in the Brador, for one reason or another, we don't know exactly why yet, we just don't see the parasite. It's not detectable. We have a million partners on this project. We hire a lot of students, some from Potlotek, some are university students. They're kind of from all over. This is my favorite picture of Anita in the world that René Lavoie had taken years ago. I just think it really captures who Anita is as a pillar of our community, and like every good parent, on Take Your Child to Work Day, I take my kids out into the field. But anyway, I do take them on those cold, snowy, icy days in case I fall or slip. So, but anyway, I'm going to keep yakking a bit about our current project, and I'll talk a little bit of McDonald's Pond, and then I'm going to pass it back to Anita. And just so everyone knows, I'm sort of I guess I'll say a bit of the quarterback or the talking head for all this stuff, but I'm really just one person at the table. I talk a lot. I like to talk a lot. I teach for a living. So it kind of comes easy to me, especially when I'm talking about the same thing all the time. I feel like Dr. Strang, you just say the same thing all the time. But at the same time, there's a whole bunch of people that you guys aren't even seeing who are involved in these projects, who just really make all this work so well. And we couldn't do this without them. But anyway, so what we decided to do, I was taught at this McDonald's Pond I mentioned just outside Myra. So I collected almost all the animals for my thesis work there. And one of the things I noticed, and I was telling Robin Stewart, who's a good friend of mine, a good friend of Anita's, that, it's weird. I go to this pond, you have high infection, high intensity, but the oysters don't die. It's not like anywhere else where MSX exists. This is just so bizarre. I don't know what's going on. And I was telling Robin Stewart this in the St. Peter's Fire Hall, which isn't far from Anita's home. And Robin said, yeah, it's really funny, that you mentioned that, because when they had the first die-offs on Phil Drinnan's lease, he was one of the first ones hit, the oysters that were really near the shore in the shallow water, like McDonald's Pond, they were still alive. It was the ones in the, 5, 6, 7 feet of water. They were the dead ones. And then Robin was telling me about Joe Gugu, who's a dear friend of Anita's. He also he was telling Robin about, he has another lease called McDonald's Pond, but it's not the Myra McDonald's Pond. There's probably multiple McDonald's Ponds on Cape Breton Island alone, I'm sure. But Joe had his lease, and he was trying to grow the oysters on the bottom, but it was too muddy. So he just said, well, I'm going to put them in cages and float them. That way, at least they'll be, that way they won't die in the mud. And lo and behold, in addition to the parasite being there, Joe's oysters weren't dying. So between what Robin saw, 20 years ago, what I was seeing in McDonald's Pond that I go to, what Joe was seeing in McDonald's Pond that he goes to, we sort of hatched this idea of, well, what if we were to try to suspend oysters across the Brador, multiple places, test, monitor mortality, monitor disease, monitor the environmental conditions, gather as much data as we can, just to try to figure out, can we grow oysters in the presence of MSX? And Joe Gugu has always felt, if you can do it anywhere, you can do it in the Brador. So hats off to Joe for having that sort of, that inherent wisdom to know that, if we try hard enough, we'll find a way. But to do this, essentially what this project is we have 14 sites across the Brador. We have 1,000 oysters floating in cages on the bottom, or floating in cages on the surface, 1,000 oysters sitting in cages just out of the mud on the bottom. And, again, we monitor for disease, mortality, things like that. So I guess one of the neat parts about this project is, I can sit at a microscope, I can drag animals in, we can open them up, we can do molecular biology on them. But where the indigenous knowledge and traditional knowledge about the Brador comes is us sitting down at Tim Hortons outside Bedeck with a map, and me saying, where do we do this? So Joe, Robin, I don't know if you were there, Anita. I can't remember now. You might have been. Maybe. Oh, I think Norman might have been there. I know someone from Potlotek was. Must have been Norman. But anyway, so someone like me who doesn't know that stuff, has to sit down with people like the Joes, the Robins, the Normans who do know this stuff and say, how do we do this? Where do we go? Where do we put these things? You guys know the places, I don't. So anyway, so that's where we have, you can sort of see, this is some of them. We've added a few since. And what we've done is we've engaged the leaseholders who look after the sites for us, because it's too many for us to look after. So, we designed the project so that they operate their lease exactly as they would as if they were farming oysters, except they're doing it as part of a 3-year experiment. So this is where we are so far. If you just sort of look at the mortality, this is a series of the leases. You can see that we have some pretty high mortality on the bottom, which is what we'd expect, and some pretty low mortality on the top, which is what we were hoping for, which is really striking. It's actually working better than we thought it would. We've had a couple of hiccups since this year. This bottom one, Dina's Pond, someone took everything. So somebody helped themselves to all of the oysters. Trying to think of Portage. We had a little hiccup there because the grower was indulging in probably 300 or 400 of the oysters on us. So that created a bit of a challenge. But you know what, that's kind of, at the time, it was like, I'm going to choke this guy. But at the same time, that's kind of the fun part of working with industry. And I think it just shows people's, that individual grower said as well, it didn't matter if they were growing or living. What really mattered is if they tasted good and if I'd be able to sell them. That's a fair point. That's not something I would think of, but that's a valid point. So for us, we're trying to rebuild an industry, which creates challenges because when we're trying to find money for research, how much is industry willing to put in? Well, there is no industry because, they've been shut down for 20 years. It's really hard for them to throw money at this. There's been a lot of money invested. So we want to see these communities and these growers get back on the water. We've had lots and lots of support from, the Atlantic Fisheries Fund, from ACOA, from Nova Scotia Fisheries and Aquaculture. I can't say enough about the people who work for, especially Nova Scotia Fisheries and Aquaculture. They have been absolute gems to work with and have helped us every single step of the way. Same with Fisheries and Oceans. The people here at in the Westmount office have been absolute gems to work with when it comes to getting licenses and things like that. So we have lots of interest and lots of support. I'm going to pass it back to Anita to talk a little bit about what, hopefully what we're seeing now means to her and her community.
Hi, those are my 2 grandsons and a nephew. But I've been teaching my community members, like elders and young people, mostly it's all volunteer work, like, off work, whatever, if they want to learn about oyster industry, what to look out for and how to identify oysters and of course, I put a price on each oyster. I tell them, listen, these are like money, like, on the lake, like, you pick them and you have a job, like, you bring them into the oyster plant. But, like I said, MSX said we couldn't do nothing. But those are the future kids. They're anxious to get back into the lake. Maybe one day they will. They're all grown up now. Like, they're in their late 20s and late 20s. And hopefully that the students get involved as well. We had some talks with the students at the high school, and a lot of the kids there, like, their parents have oyster leases. They're like, they were surprised and said, oh, I didn't know my dad had an oyster lease. Like, oh, I'm going to be working on it. Oh yeah, sure. I'll take you there. I'll teach you whatever needs to be, taught out there. Like, I'm there. I've been there for all my life. Like, I've been picking oysters since I was a little girl. So, I just have to pass my knowledge on to them. So it just continues. Like, hopefully, we have a better future for our community because we have the facility. And it's licensed, and like I always said, we're ready to go anytime. We invested so much. We got the oyster barge, we call it, like, and we have some of the boats, like the one you see right there. And that's my story. And hopefully, like, I just keep praying and hoping that we have a better future. Thank you. So I guess I won't go into all the details of this, but I think at this point in time,
what we've been able to do is collectively we've been able to move from, having no oysters growing in the Brador to, working together to find places where we, not only places where we can, but ways where we can. We've gotten the growers working together. So when you get them in a room, it's really nice. We've had multiple meetings with them, and there's nothing but cooperation among them. There's everybody, whatever differences they may have had in the past. I think the parasite's given them a bit of a common enemy to work past and get to work through. So hopefully, I agree with Anita. We're hoping that within another 2.5 to 3 years, we'll actually have some marketable size oysters in the Brador, and they can have their plant operational and selling. They already have their marketing plan set from years back. So the second thing we're going to talk about is, can I just jump right into the next thing? Is that okay?
I think that's great. I do have some questions from people. I don't know if you'd rather answer them now or later.
We can do them now, I guess, if that's fine.
As well, we're thinking about and I know people will have probably many more. We have a question from Brent, and you had mentioned you're talking about exports from the Brador. Were you talking about exports of oysters or exports of the parasite, or both?
No, exports of oysters. Yeah. So right now, yeah. So the shutdown meant you weren't allowed to sell. Like if you grow oysters in the Brador right now, if we, assuming this is going to work, continue to work, you're able to sell oysters to like Rodney's Oyster Bar or places like that, but you're not allowed to sell them to be re-soaked anywhere in Canada or anywhere actually in the world. It's because I don't know all the ins and outs. I'm trying to keep track of it all, but it's a regulated disease, a reportable disease with the Food Inspection Agency. So every year when we finish our testing, we have to send all the results to them. And so they know what's happening where. But yeah, so oysters. So for example, it's my understanding you could buy oysters from PEI, ship them to, Boston or somewhere and soak them, but you're not allowed to do that here with Brador oysters.
Thank you. And then Joe McPhee has a question. He's wondering, and this is, I think he posted it a bit earlier in your talk, what's different about the holding conditions that allow the animals to clear the infection, I think, kind of in the lab compared to in the wild?
Yeah, so that was just a total serendipitous moment where I was trying to do an experiment. It was actually the first experiment I did for my thesis. And what I was trying to do was hold oysters, or no, I didn't try. I was holding oysters at different temperatures to see if at higher temperatures, they'd have a little higher metabolic rate, to see if I could track the rate of growth of the parasite at higher temperatures versus lower temperatures. And the original plan was, maybe we can find cold refuges. So I had oysters at 0 degrees, 5, 10, 15, so hoping that, we could find cold refuges, these deeper areas in the Brador that maybe oysters could, shelter more or less or something from the Brador. Because in the US, what they find is after really cold winters, there's very, very little disease. So but I personally think that's more tied to a lack of some kind of intermediate host than it is actual. I think there's an abundance of host that disappears for a bit more so than oysters. That's my thought. But anyway, so what happened is after about 12 weeks, I said to my advisor, I said, Neil, like there's nothing happening. Like everybody's happy. Nothing. I don't have a dead oyster in this tank, in any of these tanks. So he said, let's take a sample, see what's happening. So I took a sample, and lo and behold, the oysters that were held at like 20 to 22 degrees, there wasn't a trace of the parasite in them. So they went from about 70, 80% prevalence to almost, I think, might have been 1 or 2%. So I don't know what's happening in the tank, to be honest. It's not a trade secret. It's not something licensable. But if this but it happens in the tanks, and at these temperatures in the wild, oysters would succumb to the parasite at those kinds of temperatures. So there's something in the water, whatever it is, I don't know. But again, these are all doable things. And we've, I was doing it with several hundred animals. A few summers ago, I actually did it with 4,000 animals per tank, and it worked just as well. So if oysters get the parasite, we can bring them in, give them a bath kind of thing for a week or two, and then put them back out. They'll get reinfected, but part of our desire to figure out the life cycle and detect this thing better, quicker, is then okay, we've purged it out. We don't want to put them right back into the soup. Let's find somewhere safer or not as effective, for lack of a better word, to put them. So yeah.
Yeah, and Joe made a great, so he was asking if this would purge the Vibrio species bacteria from the mussels and oysters because the bacteria don't like the cold.
Yeah.
That's really neat.
Yeah. So
And then
yeah, I don't.
Carol mentioned, is this something that you guys have thought about? Could you do this to, Carol mentions, I think that mussels are regularly purged before sale. Could that happen for oysters? Was that
Well,
something you
yes.
guys-
So oysters, you have to. So if you grow your oysters in suspended culture because of birds and everything else on the surface, you actually have to sink them for 30 days. I think it's a 30-day purge period. So one of the things that yeah, I guess there's a whole bunch of things run through our heads, but yeah, so that's one of the things that needs to happen. But one of the projects that we're going to embark on in the coming year, I'll say, is we want to find out when it comes to running a hatchery, if we treat oysters this way, if they're infected, say you have oysters from an infected area, and not that there's lots, but there are some there. What's the survivability of the larvae, the health of the larvae, if you treat them this way to prepare the brood stock versus not treating them, so there's a whole bunch of interesting projects that we're going to embark on to find, so this potentially has use not only just for purging the parasite out to, keep them alive, but also potentially as brood stock conditioning.
which would hopefully help the plant
help
get going
populate,
again.
Yeah, exactly. Yeah, says someone who's never worked in an oyster hatchery. So I'm expecting someone who knows about oyster hatcheries just to
That's
show
why
up
we
on my
collaborate.
doorstep someday.
Yeah.
Yeah.
Thank you.